Hello, everyone, and welcome to today's webinar. I'm Lauren Perry. I'm Operations Manager at Syngap Research Fund and mom to a 13-year-old with SYNGAP1. And I have the pleasure to introduce today's speaker, Dr. Julia Dahlman. Dr. Dahlman is an associate professor of biology at the University of Miami. She has 30 years of experience using animal models, including sea squirt, fly, and zebrafish, to understand neurodevelopmental processes. She did her doctoral training in electrophysiology and developmental neuroscience with William Moody at the University of Washington, and postdoctoral training in molecular and chromatin biology with Gail Mandel, and in zebrafish genetics and physiology with Paul Brem at Stony Brook University. Her research seeks to understand how genetic mutations impact the development of circuits in both gut and brain to produce behavioral phenotypes in zebrafish and autism models. More recently, she's worked with clinicians, public health experts, and mobile application developers on objective measures of gut function that families can conduct at home. A recorded version of this webinar will be available on our website under webinars on the family menu. And at the end of Dr. Dahlman's presentation, you will have the opportunity to ask questions, and we'd love to hear from you. So please write your questions in the Q&A, and it's now my pleasure to turn things over to Dr. Julia Dahlman.
Thank you so much for the introduction and for inviting me to speak.
Yeah.
I will open up the slides. I'm going to tell you today about a study that we just completed that looks at behaviors in our SYNGAP1 model, zebrafish model. And what we find is that we think that elevated arousal in low arousal environments may explain some of the hyperactivity we see in this model. And I'm speaking on behalf of the authors of this study. So Sereni Sumapipala, this was her PhD dissertation, Suha Khan, Robert Kozel, Yuichi Araki, Sham Syed, and Richard Juvenier. So this is a picture of synapse. And as you are all very likely aware, many of the genes linked to neurodevelopmental disorders are enriched at this synapse. And this includes Syngap and SHANK3, both genes that are of great interest in my laboratory and that we've generated zebrafish models for. Both Syngap and SHANK3 also share strong behavioral manifestations. So this is a paper from Leanne Warren et al. 2022 that shows scores on sensory and behavioral symptoms, where green is where the majority of typically developing responders score, and Syngap and Phelan-McDermid syndrome are out here to the right. And so these conditions are associated with altered sensory processing. And so that's something that we can really study in detail in zebrafish. So that's what we set out to do. So the central question is, how do SYNGAP1 mutations impact nervous system development and behavior? And what we're kind of trying to understand is the suite of behaviors that manifest. And in humans with SYNGAP-related non-syndromic intellectual disability, some of the behaviors include epilepsy that can be intractable, hyperactivity and or hypoactivity, disrupted sleep, increased risk-taking, and sensory seeking. So descriptions of really enjoying flowing water or perpetual motion are some of the descriptions of people with Syngap that come from these publications. So we use zebrafish in part because they have pretty high gene conservation with humans. You can do high throughput behavioral assays. So these red lines are actually tracks of zebrafish where there's one zebrafish larva per well of this 96 well plate. You can see right into their GI tracts. GI distress is also a common symptom in both Syngap and SHANK3. And they have very accessible early development, and it's possible to do whole brain imaging. So this collection of traits makes them a really nice model in which to try to understand how symptoms arise, or symptoms phenotypes arise. So I'll start off talking about what we've learned, sort of validating the model that we use. And so here is a diagram of the Syngap protein from humans, and there are 4 conserved domains: the plextrin homology in cyan, the C2 domain in pink, the RAS-GAP domain in mustard, and the quail domain in orange. And you can see that in mouse and zebrafish, these domains are conserved. And you may notice that zebrafish have 2 syngaps. So there's a Syngap1A and a Syngap1B gene, both with conserved domains. And if we look at domain conservation as percent of amino acid identity, you can see the mouse has considerably higher conservation than the zebrafish, and that Syngap1A and 1B kind of alternate on which is higher in which domain. So in C2, B might be a little higher. In the quail domain, A might be a little higher. And so so they both show high homology. In mammalian Syngap, it's very well known that there are lots of different isoforms. And this slide just reminds you of that, showing the 4 well-characterized C-terminal isoforms. So here you can see that at the 5' end, you've got 3 alternative start sites. And at the C-terminal end, you've got 4 alternative C-terminal ends. One of these encodes this alpha-1 protein that includes a PDZ ligand. So PDZ is a protein interaction domain that is characteristic of PDZ95, which scaffolds that glutamatergic postsynaptic density. And so this alpha-1 is the one that's most very highly enriched at that postsynaptic density by its PDZ interaction domain. So when we were setting out to do these studies, we wanted to understand how these isoforms related to these duplicates. And so to do that, we collected all of the Syngap1A and 1B proteins from the NCBI database, identified unique exons that they encoded, and then searched those exons against RNA databases to see which ones there was evidence that those forms were expressed. And that led us to identify several isoforms in Syngap1A. You can see that there are 3 alternative splice sites here. There's a very small alternative splice that is less than 3 amino acids. And you see that type of splice also in humans. And then these different C-terminal ends. And it gets much more complicated in Syngap1B, where there are 10 different isoforms with a lot of differential splicing at the C-terminal end. And one of these isoforms encodes this PDZ interacting domain. So that is interesting to us because it means these duplicates aren't serving identical functions. Only Syngap1B encodes this alpha-1 isoform. So knowing those things, we set out to create stop-loss mutations in the Syngap1 gene to try to model Syngap1 related intellectual disability. And this is a slide of different patient variants from Glaskamp et al. 2019. To do this, we used CRISPR-Cas9, and this is work that was done by Rob Kozel when he was in the lab. And we inject guide RNAs that target the Syngap gene and create stop codons in the N-terminal region of the protein. We validate that these create loss of function, both in larval zebrafish, where we collect RNA and we look for the level of expression of the SYNGAP1A and SYNGAP1B genes. And you can see that in both hets and homozygotes, those transcripts are downregulated, indicating that there's nonsense-mediated decay. Also, we sent some fish brains from homozygous and wild-type fish to Yoichi Araki in Richard Hugenier's lab, and he ran Western with his, they have a lot of different SYNGAP1 antibodies, and you can see that the wild-type zebrafish have a band of the same size that's found in mouse, and the mutants lack expression of that band, even though they have lots of tubulin, which is the internal loading control. So that means that we've created a loss of function model in both SYNGAP1A and SYNGAP1B. And unlike mouse, the homozygous model is viable. So we can get adults that are lacking both SYNGAP1A and SYNGAP1B. And so to study behavior, one of the things that's really well documented in zebrafish are these arousal states. So I wanted to talk a little bit about arousal before jumping into the data. And I think one of the clearest demonstrations of changing arousal states during the day is our cycles of being awake and asleep. Same nervous system, but very different behaviors depending on the time of day. And this gets much more granular. I think we all know that we need a boost at certain times of day, or we get sleepy at certain times of day, highs and lows. And these are regulated by these arousal pathways that are conserved between humans and fish, and include noradrenaline, serotonin, histamine, hyperactivity, which is also known as orexin, and dopamine, among many, many others. And so these fine-tune some of our outputs. Arousal is associated with increased locomotion. And so locomotion is one of the ways of looking at arousal state. So here what we have taken advantage of is different environments that evoke different states of arousal. So in the light, the zebrafish are very visual animals, and they're fairly relaxed, and they don't move a ton. So each dot represents a set of 5 larvae. And so this is graphing the proportion of many, many larvae that move in the light. And you can see that it's nowhere close to 100%. If you move those larvae to the dark, there's this dark response where movement increases dramatically, and that's associated with increased arousal. It turns out that just as with us, if you take away our sense of vision, you go and look for a place where you can turn on the lights again. The fish do the same thing. And then another thing that increases arousal is vibration, or tap, also known as the acoustic startle response. So you can see that the fish also increase locomotion a lot to this vibrational stimulus. And so dark and tap are both states that are associated with elevated arousal, but they're a little bit different. And you can tell that if you try to anesthetize these behaviors. So these are increasing, this is the amount of anesthesia required to stop movement in light conditions, in dark conditions, and during tap. And you can see that tap takes a lot more anesthesia to suppress that movement, in part because it's an aversive stimulus. It's something unpleasant. And so it takes more anesthesia to suppress that than either of the behaviors that are going on in light and dark. And so you can rank level of arousal from light being low level to dark being higher to tap being aversive and very high. So we started with high, we started with the tap. And to measure the tap response, we put the fish in this Noldus Daniovision chamber that has a tap device. I put a hammer here to represent it, that can deliver different strengths of vibration, which is can elicit the acoustic startle response. And then we load this chamber with offspring from wild type and SYNGAP homozygous, wild type females and homozygous SYNGAP males, and load this dish both with wild type and SYNGAP hets, and then see how they respond to this tap. And we do so this is a behavioral assay that was developed by Kurt Marsden, which is where Sereni is now doing a postdoc, and it involves 10 low intensity taps followed by 10 high intensity taps. Both of these are separated by 20 seconds. And then it goes into a phase of rapid high intensity taps in rapid succession, so only a second between each tap. And this allows us to measure how well the nervous system adapts to this stimulus, or otherwise known as short-term habituation. Then there's a recovery phase, and then another set of high intensity taps with a 20-second interval. And so what you can see is here is in the black line is the wild type response, and in the purple line is the SYNGAP het response. And basically to tap, there's no significant difference across the whole behavioral assay. Both lines have less of a move slower in response to these lower intensity taps, move further in response to the high intensity taps, habituate with repeated high frequency stimuli, and then come back to this baseline level after a rest. And so if we calculate a habituation index, which is basically this movement minus this movement, you can see there's no difference between wild type and SYNGAP hets. So this is the habituation index. We looked a little bit closer at the difference between the different phases depending on genotype, and you can see that this low intensity tap in phase 1 and high intensity taps in phases 2 and 5, the response of wild type is not significantly different across these different intensity taps, but SYNGAP has another gear. So it's at a lower response to the low intensity taps, and then has a higher response, significantly higher or faster response to the high intensity taps. If we look at percentage of response, we also see that SYNGAP is a little bit more responsive than wild type, and that SYNGAP increases its proportion that are responding as the taps get stronger. So that is, in terms of our aversive stimulus, our tap stimulus, there isn't really a dramatic difference in between SYNGAP and its wild type siblings, other than a increase in dynamic range. So what about these light and dark behaviors? We also measure the light and dark behaviors using this Noldus chamber. And we do 4 cycles. So first we adapt the fish to dark, and then we do 4 cycles of lights on to lights off. And the reason we do this is that it evokes a beautiful visual motor response. So here you can see that response. What's plotted here is the speed of movement, so millimeters per 30 seconds during these 5 minutes of light followed by 5 minutes of dark. And you can see that in the light, the speed of movement is low relative to the dark, and that it's a very robust response to dark. So you can also see, so graphed are wild type response, and then again, hets are in purple and homozygotes are in pink. So during the light, the homozygotes are most hyper, most active, and during the dark, the hets are most active. And if we plot these numbers across individuals, you can see that in light, there's a dose-dependent increase in hyperactivity as you go from wild type to het to homozygote. In the dark, there's a much subtler difference, with the hets being most different from the wild type. But we really wanted to know, so this level of analysis is pretty coarse, because this 5 minutes represents a lot of bouts of activity for a zebrafish. And so we wanted to know whether this observed hyperactivity may be due to more frequent movements, which would indicate descending commands from the brain, or increased movement per bout, which could be increased recruitment of motor outputs. And so what we did was we generated an idealized larva. And the way we did this is we had a very large N. So Sereni collected data on a lot of animals. And if you sort of pool the distribution of displacements, the size of each bout in wild type had, we had a total of 119,135 bouts across all of our 173 larvae. And by plotting these bout distributions and dividing by the number of larvae, we get behavior for an idealized larva. Normally, behavior is so noisy that you don't get these smooth curves. You only do this if you do this in a lot of numbers, so that you have representation even in these large movements where there aren't very many of those movements. And so what you see here is that the Syngap are moving much more frequently in the light. So on the y-axis is the number of events per size of displacement. And here wild type are at 500, Syngap hets and homozygotes are at 1,000 for the smallest displacement size. And in lights off, the differences are much more subtle. You can see, as indicated by the data on the previous slide, hets have the largest number of bouts, followed by homozygotes and then wild type. So in terms of this question, we definitely have more frequent movements. But seeing that stark difference between the Syngap behavior in the light, we wanted to know a little bit more about what was going on there. And so, what's happening in light versus dark? And this is data from Eric Horstick from when he was in Harry Burgess's lab. And they showed that this hyperactivity in dark is due to the fish trying to return to the light. In other words, the activity in the dark is produced by light-seeking behavior. So what does our light behavior look like compared to our dark behavior? Well, this is the probability of a behavior that has of a particular dwell time, which means the time in between different bouts of behavior. And you can see in the dark, there are lots of short dwell times, meaning they don't sit still for very long. They're mostly on the move. Whereas in the light, there are more of these long dwell times. They're more, they spend more of their time not moving. And you can see this also if you subtract these probabilities from each other. So it's dark minus light. So when you're in positive, dark is, you have more of these short dwell times in dark, more of these long dwell times in light. If you look at displacement, the size of displacement, in light you have lots of short displacements. So you have short displacements and long dwell times. And in dark, you have much larger displacements. So the nice thing about collecting these statistics about movement characteristics in light and dark is it lets us interpret what we're seeing in SYNGAP. And so here's the SYNGAP data. So in light, we have the wild type in the black circles and SYNGAP in the purple and pink. And you can see that if you subtract SYNGAP from wild type, you see a pattern that looks very much like when you subtract light from dark. So SYNGAP in the light are behaving like wild type in the dark. They're producing these kind of seeking behaviors that have shorter dwell times and larger displacements. Whereas in the dark, there really aren't differences that go along with the dose of the SYNGAP gene. So in addition to more frequent movements, we have increased movement size in the light that we're seeing from SYNGAP. And so it seems that SYNGAP are participating in the sensory seeking in the light. And so the way we're interpreting our data are that we're seeing these high arousal behaviors in low arousal environments. Sorry, environments is twice there. So here we have our arousal wedge, and SYNGAP hyperactivity is most pronounced where arousal is lowest. So what we find is that in response to these aversive stimuli, we see a larger dynamic range in SYNGAP, but not a very pronounced hyperactivity. During the dark transition, we see a greater frequency of movements, but not in a way that depends on SYNGAP het or homozygous status. And in light, we see pronounced hyperactivity where SYNGAP are moving like wild type do in the dark, and the effect is greatest in the homozygotes. So what might this mean? Well, looking at some of the literature on other things that produce these types of symptoms or phenotypes, elevated cortisol or stress hormone can produce a larger dynamic range, but it also produces freezing in the light. So we don't think anything is going on with elevated cortisol. Our data are consistent with increased goal-directed seeking behavior in the light. It's been shown that increasing these arousal peptides, CGRP or CCK, can produce increased activity in the light. Another arousal pathway, cocaine amphetamine related transcript, can produce a larger dynamic range. This peptide hormone, hypocretin and orexin, can both produce higher dynamic range and increased behavior in light. And serotonin is also associated with this increased seeking. But these are only a subset of the arousal pathways that are known to influence transitions between different behavioral states. So here are some others, and this also is not a complete list. So our central question was, how do SYNGAP1 mutations impact nervous system development and behavior? And our data really point to a role for arousal pathways. But I think the outstanding questions persist in that we have this picture of a postsynaptic density that would be found throughout the brain. So how is it that we have, so there are some circuits that seem quite significantly impacted and others that seem to be working fairly well. And so what makes some circuits vulnerable to reduced copies of SYNGAP while others are not is still an open question, and one that we're interested in addressing. So I want to acknowledge the people who contributed to this work. Dr. Sureni Sumathipala got her dissertation working on SYNGAP and And Syngap really inspired her to continue working as a scientist. So she's now pursuing a postdoc at North Carolina State University with Kurt Marsden. This work couldn't have been done without Ricardo Cepeda, who keeps our zebrafish in good shape. Suha Khan has moved on to Ellen Hoffman's lab at Yale. Robert Kozel is now seeking a faculty position. Sham Syed is from Physics, and he's the one who talked us into analyzing all those behaviors, those 119,000 behaviors, so individual bouts. Yuichi Araki did the Western in Richard Hugonier's lab, and we thank them for their support. And then this work was funded by the grants from National Institute of Mental Health and National Institute for Child Health and Human Development, and a Safari Pilot Award, and institutional support from University of Miami and Johns Hopkins. And with that, thank you for your attention, and I will stop sharing.
That was awesome. So I know who's watching from looking at Facebook and from this, and for the newly diagnosed parents, now you understand why we get so excited about models, because we just saw what happens when you can go from models to results to interpretations to so what's for the kids. And I think that's important. I have some technical questions and I have some translational questions, and then I'm happy to take questions from the audience. And I don't know, JR, if you're on the call, but I'm sure you probably have questions because you did fish once. But so translational serotonin, anything you can expand on there, because there are both sort of within the Syngap community, a lot of people have had results both with gut motility and mood.
Yeah.
5-HTP. And then within the broader epilepsy community, I think I was just at an epilepsy meeting talking to the CSO of the Dravet Syndrome Foundation, and there's 4 different serotonin drugs right now in trials for Dravet. One of which is with Longboard, which is open, by the way, to all patients with NDEs. So serotonin is something everybody sort of perks up when you say that. And I was hoping, before I go into a bunch of hopefully geekier questions, you could elaborate on what this tells us about serotonin and how we would think about it.
Yeah, so serotonin has a really interesting trade-off with cortisol in a geeky way, in that, at least in zebrafish, serotonin is associated with this sensory seeking behavior, where like they look for flowing water, and the serotonergic raphe are important for that. And in the case of so how that relates to it being an anti-seizure medication, do you know which receptors they're targeting for the anti-seizure medication?
B and C, it's different. There's different drugs going after me. I mean, I think the Longboard is the C, but I'm not looking at it strictly through the lens of seizure. Like, I mean, when those of us who are in the community are giving our kids 5-HTP religiously, we're doing it to improve motility and to improve mood.
Yeah. Yeah. So, I mean, it's a nice twofer, right? because it's, it's linked to these dorsal raphe, which are really important for kind of engaging with your surroundings in a way that is less fearful, let's say. So that's in the brain, right? Yeah. And then in the gut, serotonin, 90% of the serotonin in your body is in your gut, and is important both for communicating with the brain and for keeping the gut moving. So it's not surprising that serotonin is something that people are looking to.
So I'm taking you a little off topic here, because this is about arousal, not about gut motility, but I just want to double click on that. I have quoted you many times to people being like, 90% of our serotonin's in our gut.
Yeah.
Like one of my favorite sound bites. But we give kids 5-HTP, some gut symptoms get better, mood gets better. So we're replacing serotonin. But then if I think about it a level deeper, are human Syngap hets, and for the people who are new to science speak, hets means you have one good copy, one bad copy, and homozygotes have 2 bad copies, which are viable. It means they can live in fish, but not in human. So the humans are all hets, right? So if 5-HTP helps the human hets, then does that mean that Syngap makes serotonin and that's why they're deficient, or that Syngap hets are desperately using up all their serotonin and therefore they're deficient and therefore supplementation is helping? Like what makes sense?
Don't know. And the thing is, I mean, 5-HTP is the precursor for serotonin, but it's tryptophan, right? I mean, it's the building blocks.
Yeah.
You're getting the building blocks for serotonin.
So it could be
Those building blocks go into other pathways as well as serotonin. So I'm sorry to say this, but having a beneficial effect from 5-HTP and tryptophan could be through a bunch of different pathways. And it's something that we're interested in for sure, because of our gut work, and also because we're really interested in that gut-brain axis. But you just don't know.
That's a really good point.
Which arm?
Yeah. No, I'm glad you said that. So we don't know. There are some plausible hypotheses. There is a connection to serotonin because it's the precursor, but it also affects other things. Okay, better answer. Cool.
Also, I mean, like the bacteria break it down and make indole and talk to the gut that way, and can induce peristalsis that way. So that's an independent route to increased movement in the gut that could be via the microbes. So I'm just saying
5-HTP plus bacteria equals indole equals gut works.
Yeah.
So it could have nothing to do with serotonin.
Could bypass serotonin.
Whatever, it works. That's all I know.
That's right. No, I'm just saying we don't know.
Yeah, no, it's fair. It's fair. I'll stop saying, okay, I get it. So can we go back to the fish now? So I just, as I was sharing with you before the webinar, I just came from a meeting where a lot of people were talking about fish. And another lab that's done a lot of work with fish has decided that the 1B variant is the best model for Syngap.
Yeah.
Whereas all your slides had AB hets and AB homozygotes. So does that mean that in the hets you knocked out one copy of A and one copy of B?
Yep.
And in the homozygotes you knocked out both copies of both?
Yep.
So you're basically, so is the thinking behind having a het that's got 1 and 1 is that these two have always worked together, and as you pointed out beautifully, they have different isoforms. So basically you just mash them together and treat them as one, because the idea is that they're subdividing the job that one does in us. Which is validated by that slide you showed with the different isoforms, how they validated by that. And in data I didn't show. Yeah. It is nuts how it turns out you have to have them in balanced numbers, or you don't get survival. So if B alleles outnumber A alleles, you don't recover as many. If you do a het incross, right? I mean, that's kind of nitty gritty, but there's an interaction between those two. And there's data from mouse as well. If you just knock out alpha-1, that's a really hard hit for Syngap. Like, that's worse than knocking out the whole gene. And I think our data are consistent. It's a different hit, if that makes sense. So that, it makes sense. And it takes me in two different directions. And then we'll come back to the title of your talk. The first direction it takes me in is if you were doing a drug screen with zebrafish, based on what you know, and you know a lot about Syngap, you probably know more about Syngap zebrafish than anyone. What model would you use? Would you use an AB het? Yeah. Okay. And then I want to double click on what you said about alpha-1, because for the audience who are like, where is Mike going with this? There are different therapeutic approaches for Syngap that people are talking about at the genetic level. One of them is something, you have a good copy and a bad copy. One of them is ways to make the good copy work harder and make more. The isoform problem goes away there because the body decides which isoform the good copy will turn into. Other people are talking about replacing a specific isoform. And some of these arguments are, well, look, we know alpha-1 does a lot, so let's just throw in alpha-1. And some people are like, well, you don't know what the other 10 are doing. That's where I sit. That could screw things up. And so maybe we should think harder about that. Now, if I think about what you just said, that sort of validates this be careful picking a horse until you know what the other horses are doing argument, right?
Yeah,
Interesting.
very much so.
And then you could also take what you said and spin and argue the other way, right? You said if we take out alpha 1, it's worse than taking out the whole gene.
That's right.
You could say, well, maybe the converse is true. Maybe if we just put in alpha 1, we're fine.
You could argue that way, but I'm telling you, the balance is important.
Yeah.
The survival data is they're very clear. And honestly, you know, SYNGAP1B mutants clearly survive fine if that's the only horse in the game. If that's the only fish in the game, it's fine. But if there are other genotypes vying for resources with that SYNGAP1B homozygotes, like we didn't recover any from that het-hitting cross. We had like an N of 131, right? And no SYNGAP1B homozygotes with SYNGAP1A wild type.
We're viable.
So it's just that's one of the reasons that we think. And you know, I mean, I actually haven't looked at. It seems like a lot of the patient mutations are happening in the middle of the gene, the part that isn't alternatively spliced.
Right.
So that's going to affect all isoforms.
And there's at least two then, and if you're going to go down that rabbit hole, like it said in Vlaskamp, and we've seen it in the patients, people with mutations in the first 4 exons don't seem to be as, they're disabled for life, but they're not as severe, right? It's
Yeah.
Step function. Similarly, there's this other mutation at the end, in the intron, where in another model, it is a het, but the balance of isoforms is different. And the human version of that, at least the younger ones, have another step function increase in function, right? So there's a lot going on with the splicing, but you're right. Anyone in the middle with a protein truncating variant in the middle is probably a het. And what's weird about in the clinical world, we used to get diagnosed post-seizure, right?
Yeah.
So all our kids had seizures. Now, thank God, doctors are seeing delayed kids and sequencing. And so I have the unfortunate role with, I've been on the phone with parents of 1 and 2-year-olds, and mom is holding this beautiful little child where the phenotype isn't very obvious yet, saying, well, maybe we're going to get lucky. And I'm looking at the genetic report and I'm like, I hate to say this, but I doubt it. Like, you need to know what an absence seizure looks like, what a myoclonic seizure looks like, and be ready, because we've got a protein truncating variant right in the middle of the gene. It's coming, baby. And the sooner you identify it, treat it, the better.
But so that's the other thing that is, you know, to bring it back to arousal.
Yeah, please.
This, you know, Sullivan, Kadam, there are papers that have looked at seizure, and those seizures tend to happen at state transitions, you know, between REM and non-REM, and between wake and sleep. So, and those are arousal transitions. Hi.
This is John.
Hi, John.
He tends to demand my attention sometimes. I'm very familiar with the Sullivan paper. And John, you're embarrassing yourself on video. And that's a really good point. I thought of it. I've always talked about that paper in terms of wake, sleep, and sleep, wake, but you're talking about it now in arousal states. And I thought the two things you had on that slide, on that presentation that stuck with me, were the 2 wedges. So with low sensory input, they're seeking it more, right?
Yep.
That makes sense. So it suggests they need a lot. But then if you go to the tapping, when you give them too much, they tend to get tweaked out.
Well, the difference between with wild type is much less pronounced. It's just not pronounced.
Yes, understood. And John, be chill. And then the other thing you said was that I liked it when you connected the curve between SYNGAP minus wild type to dark minus light.
Right.
That was like okay, that makes sense. So, and that's the title of
Yep.
John, apparently ate something bad yesterday, threw up, so school won't let him come. He's completely fine. He's making me nuts, but I can't send him to school right now. So that's why John is with us, in case I was wondering. So what can you, so like, is the takeaway light, dark minus light equals Syngap minus het means what?
Syngap minus wild type. It means that Syngap is so in dark, the fish are seeking light. So what we think it suggests is that in light, Syngap are still seeking. They're still doing this sort of sensory seeking behavior, even though they're in a context where normally that would have fish just being chill and not doing much. So they're behaving as if they're in dark, and behaving as if they're in dark is behaving like you're seeking sensory input. You're searching for something.
I think we should study zebrafish siblings of Syngapians and see if they learn to create more stimulation as a result.
Is that what happens?
Yeah, I think that might be a viable hypothesis. But does it suggest that? Okay, here's my last question. Does it suggest that more stimulation will satisfy them, or does it just suggest that they are insatiable, that they cannot process that sensory input without some kind of chemical aid?
That's a good question, and we need to look at that. It's so the data make it look like the hyperactivity is most pronounced in the first period. So they're doing this seeking, but you repeat, and finally it goes down. So we need to look at that a little bit better. And what we'd like to do, I mean, you know, when you asked, what we'd like to do is test a bunch of the seizure meds and a bunch of the gut meds against this range of phenotypes. So gut and arousal and see whether there's something that does do what the serotonin pathways have done on a sort of case-by-case basis that can improve both.
Just bribed John's complicity for just one more minute, and then I almost could. But so two thoughts on that. The first is we have done a drug screen with Clement Chow in flies.
Mm-hmm.
That data is publicly available, and we're happy to share it with you if you're thinking about drug screens.
Sure.
Also doing a drug screen with rare base,
mm-hmm.
where that is protected by various agreements, but there is a short list of molecules in a couple of classes there. But might be worth testing in a model that we can't talk about publicly, but we probably talk about privately. Okay. So if you're heading in that direction, it would be really interesting to look at those. I think. I love you too, John. Because I mean, and I'm going off topic, and then I want to defer, and then last call to the audience. I do see we have Professor Courtney in the audience, and Yulia and Marta and others. So if anyone's got a comment, please ask it or text me. But I'm glad to hear you're thinking about testing drugs against phenotype in fish. And, you know, I always say this, please call us if we can help support that work. Because I think it's really important. And what I'm starting to appreciate when I started SRF is we need a genetic therapy. And now, you know, guess what? We're blessed. We have companies publicly and privately working on really cool technology.
Yeah, that's awesome.
But we're cursed in that the FDA is still figuring out how to approve this stuff, and companies are still figuring out how to apply it. And it's going to happen, but it's going to take too long. So my passion and focus on repurposed drugs is only grows with time. And I'm really excited to talk to you about anything we can do to validate.
Yeah, no, I think I'm very excited about it too. I mean, like you talked to, so I am right down the street from Andres Jimenez Gomez. And, so talking about the seizure meds and the pathways they work through, it's really like it's all over the map, right? And having a way to empirically test them against the idiosyncrasies of a genetic condition, I think is really helpful. So I will look up the Clement Chow stuff, and and I'd love to get your take on those things, because we're now that we've looked at a number of different outcomes, right? We've got ways of looking at the gut. We've got ways of looking at these arousal pathways. I think we're ready to screen. We can interpret what we get, if that makes sense.
No, it does make sense. And Chow will be talking at our meeting. So if there's any way you can make it to Orlando that day, it would be great. He will be there in person. Thank you so much for doing this webinar.